Okay, so today, inspired by the book of the year, The Immortal Life of Henrietta Lacks, I'm here to share with you some history and also some thoughts on where growing human cells in culture is taking us. And it's really interesting, really promising, but it also brings up some questions about responsibility and the fact that we need to have knowledge shared across all people so that we really make educated choices and understand what's going on and what's coming. So really, I'm going to briefly talk about tissue culture, introducing the idea of what cells are and where they come from and what they turn into, but then really talking about some future applications that are really right around the corner. So first, what is a cell? Well, a cell is the functional unit of life. That is the smallest unit of life. All living things have one or more cells. And in the simplest terms, a cell has a barrier that we call the cell membrane, cytoplasmic membrane, or plasma membrane. It's a barrier. Keeps things in that belong in, keeps things out that belong out. In the simplest terms, that's what it is. The cytoplasm. The cytoplasm is the sort of viscous filling. It's got organelles, and it's got all sorts of factors in it that interact with each other and conduct the signaling that the cell requires to accomplish what it needs to and to respond to its environment, to produce energy in a usable sort of currency. All of that stuff is contained in the cytoplasm. And then, of course, you have the nucleus. The nucleus houses the DNA. And if you didn't know, the DNA is the blueprint of life. And it contains the instructions for how to make everything we need to have life. It gives us our unique features. It allows the cell to accomplish everything and build all of its structures. And it's a very, very long, skinny molecule, and the sequence is contained right in the middle of it. It's long been a hope and an exciting sort of prospect for scientists to be able to not only understand how DNA is organized on a genomic level, and when I say that I mean all of the chromosomes and the addresses for what belongs where, but also to be able to manipulate that. And not just manipulate it in an irresponsible sort of willy-nilly way, but in a way where we can really improve the human condition. Curing disease, reversing aging, things like that are actually right around the corner. So to put in perspective what we're talking about today, I thought it would be really nice to have an idea of the size of things. We often overlook these simple things, but really paying attention to that gives you an appreciation for how scientists have to consider what they're doing and the tools that we need to use. So starting up at the top, and here on the y-axis you've got a logarithmic scale, so every unit is tenfold increased in size. So here we've got the human body, okay, human height roughly, a little over 1 meter. And as you move down, you can see, oh wow, the human oocyte's quite large. Muscles and nerve cells can be quite long as well, but the oocyte's quite large. And then red blood cells are one of the smaller cells in the human body. But mostly the cells we're going to be culturing fall within this range right here, between 10 micrometers, which is 1/1000th of a meter, or 10 1/1000th of a meter, up to 100 micrometers. So that's really the range of size that we're talking about here today. And when you consider the human body, that's at an organismal level. And the organism is made up of various organ systems. Organ systems do particular jobs in the body, and they are collections of organs that do those various jobs. The organs are made up of tissues. The tissues create the architecture and organize the cells that are specialized to do the jobs that allow the organ to do its job for the system. Cells are composed of many, many molecules, and molecules are made up of atoms. So I've enlarged cells here so you can see exactly what we're talking about today. And this is looking at the cellular level, and then collections of cells that are tissues. So cells are specialized. They have to do a particular job in fitting in the tissue of the organ system of the body. So we have all different kinds of cells. We have cells that cover our surfaces. So for example, the skin cells, okay, the epithelium of the skin. We have linings like you find here, okay? Linings in the mucous membranes, all sorts of different linings. We have storage occurring, for example, in the liver, where we have storage of sugars, okay, and fats too. Movement, we have muscle cells that can contract and accomplish movement. We have connective tissues, the cells that make the connective tissues, achieving the connections between tissues. We have defense cells, some of my favorites because I studied immunology, so that's where we get defense from. And here you can see a lymphocyte. That's one of our defense cells. We have cells that communicate. In fact, communication, we're seeing here a neuron, and boy, do they communicate. But the truth is that all cells communicate with each other. They communicate with each other by excreting things, sending different molecules out that act as sort of a voice that say, hey there, how's it going? Things are rough over here. I've got an infection. I've just detected something that doesn't belong here. I'm sending you an alarm. Okay? It's a virus. Get ready. Okay? They send all kinds of communication signals. They shake hands. They have molecules that interact with each other and go, hey there. They're called cell adhesion molecules. So that's communication. So while we think of things like neurons communicating on a grand scale, they certainly do. But really all cells communicate in some way. And then, of course, we have reproduction. That's the most exciting part of human life, right? So we have the sperm and the oocyte. So those are very specialized cells. And all of them come from one cell in the very beginning, the fertilized ovum, right? The zygote. So let's look at that really briefly. And I know this can get very complicated, looking at this whole cycle of life. But it really helps to appreciate where cells started and the changes that have to occur in order for them to become useful to us. Therapeutically, clinically, research-wise. So, of course, a man and a woman join their egg and their sperm, and you have fertilization that occurs. That's the beginning, right? And then when we're looking here, we can see the pre-embryonic period. And what's going to happen at this point once the oocyte is fertilized is rapid cell division. Cell division after cell division after cell division. And you have this number of cells. So the size doesn't necessarily increase, but the number of cells does increase. And they begin a process called compaction, where they get smaller and smaller and sort of collect on one side. And it leaves behind a sort of fluid-filled pocket. This is called the blastocyst. The blastocyst is what implants in the uterine lining that a woman prepares, a woman of childbearing age prepares every month. That's the menstrual cycle. And so it embeds itself in this very warm red blanket, okay? And it begins to implant, and that happens at about 7 days, okay? So between here, 7 days, and this line right here represents 2 weeks, 14 days, you start to see these purple cells right here. They differentiate further, and I'll show you another slide in a minute. And you start to get a process of gastrulation, okay? And changes that begin at this particular point is where cells become what we call fated. They start to become limited in what they can develop into. That's why I'm telling you all about this, where it all starts from and where we're going, okay? So at this particular point, there's limitless potential. So when you think about something like identical twins, okay, monozygotic twins, that's where that happens. Those cells have the potential to become an entire human being, okay? So we say these are totipotent at that point. Toti, they can become anything, okay? Potent, they're potent, they can become anything. And of course, when we talk about these things, people always ask certain questions. And so here's some important ones. Day 7, implantation of the blastocyst. That's where it embeds itself in the uterine lining, the endometrium. Day 8, that's the last day identified where there can be a complete separation of monozygotic twins. Generally speaking, if the cells split off after day 8, Siamese twins develop. Okay? Or something where one of the twin dies, a parasitic twin develops, something like that. Day 14 is an interesting time. Day 14, at the conclusion of the 2nd week, and we're talking about the 2nd week of embryonic development, of human development. We're not talking about gestational weeks. I know we have a lot of women in here who may have had a child, and when they track a pregnancy, they'll track it with an extra 2 weeks built in, because the 2 weeks before fertilization count when you're looking at gestational age. So when we're talking about, the developmental age from fertilization, we're actually talking from fertilization, okay? So 7 days after fertilization is when implantation happens. 8 days after fertilization of the egg is when you can't, have monozygotic twins develop after that point, identical twins. And day 14, 14 days after fertilization is when you have the development of a structure called the primitive streak. Once the primitive streak develops, the cells, that's when we really start to see the cells become limited into one of 3 what we call lineages, okay? And these come from the 3 embryonic germ layers. So that's when this happens, okay? And now, while we're looking at implantation and the uterus, I just want to take a moment to talk about Henrietta Lacks and really make sure everyone's on the same page of understanding where her cell line originated. Her cancer was on the cervix, okay? So this is looking at the base of the uterus at the end, okay, of the vaginal canal. So that's where her tumor was, and that's what they took. And so you can also appreciate the fact that this was a very intimate, any cancer is intimate, okay? But this was a very intimate cancer in that way as well. I think that's something to appreciate. So when we look at the development at this point, we're looking at 14 days at this point, right here, okay? So what you can see is the epiblast cells, which are depicted in purple, and I didn't point those out earlier, but I'll just tell you that's where the human embryo develops from, okay? And this is the beginning. This development of the primitive streak is the beginning of that differentiation that will become the full human body, okay? So the primitive streak occurs as cells move, they migrate. Do you remember I told you all cells communicate? And as they communicate, they send signals out. They actually spit things out that other guys can hear, but they also shake hands. They go, hey, I'm going over here, you're going over there. All of the collective action of those communications cause the cells to start making certain gene products, but also stop making other gene products. It's this change that occurs that yields differentiated cells. It causes them to change. They become more and more limited in their potential. We say they become fated. So through human development, we have the start where we have totipotency. That cell can become anything. But as soon as gastrulation begins, at the beginning of the primitive streak, once the cells start migrating and forming the 3 embryonic germ layers, which you can see here, okay, ectoderm being the outermost, mesoderm, and endoderm. This process right here is where you start to see real true limitations coming out in the formation of the 3 germ layers. And all of the tissues of the adult body can be traced back to one of those 3 lineages, okay? So when we talk about embryonic stem cell therapies or embryonic stem cell research, ideally we're talking about a researcher working with a very early totipotent cell, and I've heard people say pluripotent. Pluripotent means it can become many, many tissues, okay? So there's a range of potency is what I'm trying to get across here. And it becomes more and more limited as they become differentiated. So the differentiation of cells, how is it they become limited in their potential and fated to become one type of tissue? There's interactions with hormones. There's effects that can be environmental in nature based on their placement, where they are. Is there another cell on the other side, or are they by themselves? Gene regulation, cell interactions, all of those things impact its changes, the changes that occur to it. And so as a researcher, somebody has to actually explore what features are making it take a certain turn in the pathway of all of the things that cell can become. And it was thought for a very long time that you could never go backwards. But I'm here today to tell you that actually you can now, okay? So there's been incredible leaps forward in stem cell research that have now brought us to a point where you could take adult cells and go backwards. So we're going to talk about that. So taking cells from the blastocyst earlier on, researchers can take these and culture them. This is culturing. These techniques, culturing cells, are really largely due to the fact that there were cells like HeLa long ago that everybody could use and that grew so readily that they could explore all different sorts of media combinations, all sorts of different incubation possibilities, temperature and carbon dioxide percentage. All of these things got, sorted out using cell lines that grew so aggressively, really. And so we really owe that to the earliest cell lines and the people who they came from. The researchers that did the work, but also the people that they came from. And so these cells can actually be forced to differentiate in a number of ways based on all sorts of research that's gone on. So I've explained to you that cells differentiate. They start out totipotent, they become more and more limited, ranging from pluripotent to eventually in the adult human body, they can actually become terminally differentiated. Terminal means the end, right? So terminally differentiated cells don't divide. They have a function, and they are very specialized, and they do that job, and they don't divide and make more terminally differentiated cells. There are actually stem cells that are maintained that replace many of the terminally differentiated cells. In fact, they replace a lot of the ones that we never thought could be replaced. So that has been a sort of up-and-coming idea as well, is that there actually are some stem cells in the adult human body that are maintained as such. So to give you an idea of cells that have rapid cell production, and I hate to say this, but if you've ever had chemotherapy or know anybody who's experienced chemotherapy, you probably have a pretty good idea of what tissues are affected, because chemotherapy harms the most rapidly dividing cells. The idea behind it is that when we use chemotherapy, we kill off the most rapidly dividing cells, which should be the cancer, right? But actually we have rapidly dividing cells in the adult human body, like our skin, in our mucous membranes, hair, nails, and our blood cells. All of those get affected by drugs that kill off rapidly dividing cells, like, for example, chemotherapy. So we've known that for a long time, okay? There are many cells that have little or no cell division and replacement going on, okay? So places like the liver, the kidneys, the eye, the muscles, and certainly the neurons. In fact, we never thought neurons had we never got any new neurons, but our understanding of that has also changed. There's a small amount in one particular part of the brain, right? Chris could probably point it out. And there's a small number where you get occasional new neuron growth, but it's dangerous to have neuron growth in an encased bone. You can't have a bunch of new cells being built in a limited space. So that's dangerous there. The liver, you might say, really? There's no cell replication there? Well, yeah, the liver can be regenerated. A lot of people are aware of that. But it doesn't typically you don't have cells being made a lot unless there's some damage being done, okay? So this chart I put up here is actually really fascinating. This group, bionumbers.org, they went and they looked in the research, and using, I believe it was C14, radioactivity, so with atomic bombs, okay, that have been tested in various parts of the world, there are small amounts of radiation that have moved throughout the Earth. And so there have been little changes that you can detect, radioactive isotopes, in human tissues. And you can follow those radioactive isotopes and determine age, right? You've heard of C14 dating and stuff like that. So you can actually look at the cell types, how often they get turned over. So this little turnover time gives you an idea of cells that get replaced more often and cells that virtually never get replaced. So every female, for example, is born with her oocytes, and that's how many she's going to get. They're already set aside in her ovaries, and that's that, okay? For example. So this kind of gives you an idea, and here's references if you want to look into any of those, okay? So cell division, the ability of a cell to actually divide and become more than one cell, okay? It's very regulated. In order for it to happen in a healthy way, the cell has to build up all of its supplies. It has to copy its DNA with fidelity. You want to make sure the DNA is transferred from one generation of cells to the next with complete fidelity, the same order, exactly the same sequence, precisely the same. You want to have identical daughter cells to the parent cell. So it's something that has to be done with the right timing, and it has to be under control. So you can't have the cell dividing willy-nilly without the DNA having been finished with its replication, or polymerase, the DNA polymerase that makes the new DNA, rushing through and ignoring any proofreading errors that it's made. That would be bad. That's dangerous, okay? So that does happen. It shouldn't, but it does, okay? It's very tightly regulated, but sometimes things go wrong, and when that does happen, we can develop cancers. So cancer, I have to bring up, because of course that's how the very first cell lines were established. So some hallmarks of cancer that were published by Hanahan and Weinberg, they published a paper in 2000 that's been very highly referenced by all sorts of cancer researchers. And they sort of pointed out some key features that you can identify in cancers. And the first is that the cancer cells support their own growth. They're sort of like patting themselves on the back going, keep going, guy, keep going, okay? And then they resist inhibitory signals. So even though they might get some sort of message like, hey, cool it, okay? Or you're not ready to divide, they ignore it. They resist apoptosis. Apoptosis is cell suicide, okay? Or we also call it programmed cell death. Sometimes when damage happens, cells are told, you gotta go away. You're bad news. You're a problem, okay? And that's an important feature that happens. That's actually what allows us to have fingers that aren't webbed. When we were developing in utero, all this was connected, and those cells in between were told, hey, you need to go away. You're not a frog. Okay? So that's apoptosis. Cancer cells can say, no, I don't want to. I'm going to keep going. They can multiply indefinitely, and this is how they became the first cell lines, human cell lines, okay? And certainly at that time, researchers were trying to culture normal human tissues, healthy human tissues, but they couldn't because the cells had a limited number of divisions once they put them in the broth. They put them in these flasks, in the media, put them in the incubators, gave them everything they possibly could to support them, and they would undergo a process called senescence, where they age. It's effectively aging, and the cell goes, I'm old, I'm done, I'm not making babies anymore, okay? But these cells don't do that. They keep going and going and going, and that's why HeLa is all over everywhere in the world right now, okay? Because those cells kept dividing, and her tumor was a particularly aggressive one. So those ones grow like crazy. So we used these to develop cell lines. They also stimulate cancer, again, stimulates the development of new blood vessels. So I put up this picture here of Judah Folkman. Judah Folkman should have gotten a Nobel Prize, okay? But he was amazing. He basically, with much criticism, forced the idea out there that cancers need to be fed. Like any other living thing, they need to metabolize. They have to take in nutrients, and they have to put out their wastes and have it disposed of. They need those same things that other cells need, particularly because they're so metabolically active. He also identified the fact that tumors have angiogenesis going on. That's how they get the blood coming in that could bring nutrients, that could bring oxygen, take away carbon dioxide, take away, wastes, metabolic wastes. And then he also helped to identify the first angiogenesis inhibitors, drugs like Avastin, for example, that can be used to stop the growth of new blood vessels and sort of choke to death a tumor and go, you're not getting any blood, okay? That was Judah Folkman, really awesome guy. I had the pleasure of having lunch with him in grad school. He was really a nice man. They invade local tissues, but they go out into lymph nodes and they spread to distant sites. Hopefully you don't have any familiarity with this because it's scary stuff. It's called metastasis. And that's what we really worry about with cancers, that they spread out and become more difficult to treat and stop. They create abnormal metabolic pathways. They're moving so fast, they're doing all kinds of stuff out of whack to build energy and do all kinds of things. They evade the immune system. So there's some sneaky ways that this happens, including hiding what they're doing. Every cell in the human body puts up some information about, this is what I'm building in my factory, okay? And we have white blood cells that surveil that process and go, hey, what are you doing? What are you building in there? Yeah, okay, you're all right, okay? But they hide that. And there are other white blood cells that can come along and say, you're not showing me what you're doing, so you're done. But they can prevent some of that as well. So they hide from the white blood cells that would otherwise take care of getting rid of cancer. Having a healthy immune system is one way to prevent developing cancer. Genome instability. What does that mean? As these cells divide so rapidly, they acquire more and more and more DNA damage. Because the DNA has to be replicated so fast to keep up with the rapid division, and the division's happening too soon. It's not controlled. And so more and more DNA damage accrues. In fact, in one particular cancer battle somebody may be fighting, they may have cells with different genetic material inside of them because there's genetic instability. Which is also why when you're reading that book about Henrietta Lacks, you may have read the part where it says, when they took the cells first from the tumor and then they developed clones, they separated one cell and grew up one clone, and it had characteristics that differed from other ones. That probably has something to do with it, because there's variation once these cells become unstable. Inflammation. When you have these rapid cell divisions and moving into other territory, choking off other healthy tissues, it's natural to have some inflammation that develops. So all of these things are considered the hallmarks of cancer. So the cell lines of the past, to kind of go back to tissue culture, came from cancerous tissues for all of those reasons. They were aggressive. They wanted to grow, right? Whatever the case. And so they were easier to grow, if you will. That's not to take away from the work that those researchers did. It was hard to get it working, but they were the easiest. How about I say it that way? They were the easiest of the cells to first get growing in culture. Basic science findings that were made from these cells were revolutionary. All sorts of techniques were perfected based on these cell lines that could allow researchers to have easy, ethical, repeatable human cells to experiment with. Human cells to collect genetic information from and compare to animals and compare to diseased states, compare to healthy states. All of this was revolutionary. And it opened up the potential also to the possibility of growing human biomolecules in cells and collecting away from those cells things that could be used as therapies to help people. This right now is a momentous time in biomedical research and engineering. It has been for quite a while, but I feel like it is way more now than ever before because we're on the precipice of something massive, okay? And it kind of reminds me of one of my favorite quotes. So this is actually from a letter that Isaac Newton, the man who identified and proposed the theory of gravity, right? He wrote to Hooke, who was part of the Royal Society. He said in this very lengthy letter, "If I have seen further, it is by standing on ye shoulders of giants." Now he was referring to the fact that he was standing on the knowledge that had been presented to him from all of the scientists that had come before him. What a humble guy. Right? He arguably is like the greatest scientist ever. and here he is giving praise to all of the scientists who came before him. But I want to use this quote to go a little bit further and say that not only are we standing on the shoulders of scientific research giants, but we're also owing a debt of gratitude to all of the people whose tissues we have used, including Henrietta Lacks. What a massive contribution she's made. And somebody who is humble and from a humble situation and wasn't given proper notification of what her cells would be used for, we owe her that gratitude. So, and I don't mean to suggest we're standing on her shoulders, but we're certainly benefiting from the knowledge gained from her sacrifice. So now, where are we going? Now, we're at the point where you can take cells from an adult human, okay, and you can revert them back. So when I showed you all of those slides about human development, early human development, pre-embryonic and embryonic development, and I said, you know, the understanding, the general idea was that we couldn't go backwards. That's not true now. We understand that's not true. We can go backwards. And we now understand that due to a lot of work being done by a lot of scientists. And in fact, I'll show you in a minute a couple who earned the Nobel Prize over their work. So now we can get cell lines. Cell lines are cells that we can grow indefinitely in culture, and we can share them, much like how HeLa's cells have been spread around, okay? So we can now develop cell lines from all different kinds of tissue, from all different genetic backgrounds, from all different potencies, and I'm referring to totipotent, pluripotent, terminally differentiated, like all sorts of different cell types. Disease states, from diseases that perhaps haven't been studied as much as others. And also differentiation states, where different tissues they come from, different capacities these cells have. We can grow a lot more of these and tinker with them more. We can take stem cells and purposely differentiate them into these different sort of types in order to study them. It can now be individualized. The possibilities for producing individualized therapies is greater than ever before, and in a way that probably not many people could have imagined was not science fiction. This isn't science fiction anymore. So what lies ahead? Number one, I'm going to introduce the concept of the induced pluripotent stem cell. Induced, it wasn't pluripotent in the adult body, but we can take an adult stem cell and we can turn it into a pluripotent stem cell now. We can avoid working with human embryos, which of course, whatever your opinion on it, is controversial, okay? It's something that gets battled all over the world. And so if there's a way that we can do it that is actually better for therapy potential, because we're using an individual's own genetic background, it's kind of nice, okay? Avoid controversy, work freely with a readily available tool. That's much easier for a researcher than fighting legal battles. Grow any type of tissue. You can take these pluripotent stem cells and you can purposely differentiate them into the lineages that you want. We still have exploration to do to perfect it, but we're getting there. The fact that we can go backwards and create induced pluripotent stem cells is the first step. Controlled gene editing. Unlike before, we can actually program a nuclease, an enzyme that cuts DNA, and we can tell it precisely where we want it to cut. That is revolutionary. Anybody who's familiar with something like polymerase chain reaction, that was revolutionary. That completely changed everything in molecular and cell biology and biomedical research. It gave us the ability to improve forensic science as well. It's massive, okay? I can't overstate that. This is on par with that. We're going to be able to cut the genome and insert things that make it better. So if somebody has a congenital, a genetic disease, we can go in and go, that's defective, snip, put in the good one, okay? So we're talking about the possibility of designer babies. And it was actually just in the news this week that a Chinese group, and I didn't put it up yet, I literally saw it yesterday, I didn't put it in the slides. Just yesterday, a Chinese research group took human embryos, they took fertile oocytes from women who donated them, they inseminated them or fertilized them with sperm from men who carried genetic diseases. And then they attempted using this technique that I'm going to play a little video for you on, and they were able to revert that to a healthy version of the gene and actually eradicate the disease gene. Okay, that's awesome, but wow, what responsibility. Do you want to have bat wings? I don't know, I'd be kind of cool. Do you think somebody in the world somewhere would be like, I want bat wings? Or actually, let me put it in a more likely perspective. You're Kim Jong-un, okay? Do you want to build a massive army of ultra-muscular, brilliant, loyal followers? Yes. Dictator. It's real. It's real. So you can say we're not going to do that. You can say that's wrong, but there's always going to be somebody who has the power and the sickness to try and do something like that. Okay? So this is something everyone in the world needs to be aware of, and it's also something that requires diverse representation so that it's handled equitably. If the book of Henrietta Lacks and the history that was provided in that book didn't share with you enough about the Tuskegee Project and other things that happened to minorities, we need to be aware of this, okay? And we need to protect people. So controlled gene editing, revolutionary. And lastly, I'm going to show you a couple things on tissue engineering and regenerative medicine. And these involve the use of scaffolds that we can then foster the growth of cells on these scaffolds that can then be used to replace particular organs in the body. Amazing. So you've heard of people say, oh, I'm going to grow myself a new organ. Yeah, that's actually around the corner. That is going to happen eventually, okay? So first, let's start at the beginning. Dolly and Polly, we've all heard of the cloned sheep. Now, the scientists that determined they would try to do this actually used some earlier work that was done on frog eggs. And that actually won him the Nobel Prize, the scientist who did that. So he determined that you could take a nucleus from an adult frog cell and put it into a fertilized egg. Take out that nucleus and put this one in, replace it basically, and that it would revert back to a beginning state. It would go from being an adult nucleus and it would develop into a frog. That's amazing. That was our first sort of perception that, whoa, you can go backwards. At least frogs can. Okay, frogs can. So maybe humans can, right? Maybe we can get there. And so now, of course, we're there. Now, when they did this with Dolly, this was a huge leap because it went from, oh, wow, you could do that to frogs? That's nice. They're frogs, okay? To, oh, my God, you can do that to mammals. Wow. We're mammals, okay? So that really opened the door to, we can do this. It just takes somebody to try over and over and over again. But eventually we'll get it. And so that's where we are. So what does it take? It takes an adult cell. They use some mammary epithelium from this older sheep. And they took an oocyte. They took a sheep oocyte that was ovulated, and they added the nucleus from the adult cell, having removed the nucleus from the oocyte. And then they implanted them in a sheep who was willing to be pregnant, okay? and she happened to grow the ewe, and there it is, a clone, okay? So again, this was the first evidence that this could happen for mammals. So now, Kazutoshi Takahashi and Shinya Yamanaka in Tokyo, they listened to this and they learned from this and they go, hey, why don't we try and figure out how to do this with some differentiated, so again, limited in potential, adult human, well, actually they didn't do human yet, adult somatic nuclei, Can we take those nuclei and put them into oocytes? They were working with mouse first. Can we put those into some oocytes and get a mouse to generate from that? Can we reverse this for mice as well? And can we, if they can do that for mammals, can we do this for an adult cell altogether? And yes, they could. And how did they do it? They didn't move the nucleus. If I said that, I'm sorry. They didn't do that to the nucleus where they move it into the oocyte. They actually put these factors into the adult cell, replicating what you find in an oocyte. So there was no oocyte necessary, okay? They basically got the expression of OCT3 and 4, SOX2, KLF4, and c-Myc, blah, 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 okay? Jargon, right? Those are called transcription factors. And those are things that can go and bind to the DNA and change the expression of various genes, making certain genes turn on and certain ones turn off. And what they accomplished was that these cells went back to being more potent. Even though they were differentiated adult cells, they were able to bring them back to a state where they could become all different kinds of tissues. Now, at that particular time, that was revolutionary. That was 2006. And people went, whoa. But they weren't perfectly like embryonic stem cells. They weren't perfectly pluripotent like those ones were. And so some more work was done, of course, because this opened the door to the possibility, and people all over the place started working on it. And so Thompson was one of the scientists in Wisconsin, and Nanog became something that he identified, and also Yamanaka identified this as well. So that when the cells started making this protein called Nanog, whoa, those particular cells were actually just like embryonic stem cells. So now we're at the point where we can take adult cells and using these factors, revert them back, reverse the clock. Wow. So aging is a question now. Is aging a disease? People think aging is a disease. A lot of scientists think aging is a disease. And now this sort of prepares the possibility that we can reverse aging. Okay? If we can harness this technology and deliver it through the body, it's something that could reverse aging. So this is Yamanaka down here, and he won the Nobel Prize for this in 2012, along with Sir John B. Gurdon. And John B. Gurdon is the gentleman who worked on the frog eggs that I told you about. So he sort of opened the door in the first place to the fact that we can take adult cells and bring them backwards into an embryonic sort of state. Very exciting work. So what kind of possibilities are there? Well, we can take adult cells, revert them back to being stem cell-like, and differentiate them into any tissue we need, creating an autologous transplant, meaning we can replace your tissues that are diseased and damaged, and I'm going to show you an example of that in a minute. Programmed, genetically modify them. We can change the DNA in them if somebody has a genetic disease, and we can fix it and replace some of their tissues, or perhaps implant them. So something like cystic fibrosis, people have defective genes. If we could get cystic fibrosis expressing cells and implant them in their lungs and get them to take, perhaps we could reverse that disease. Okay? Amazing possibilities. Incredible drug screening opportunities exist. This is something that would be overlooked unless you're in drug screening. But the truth is that when somebody's trying to, come up with some new pharmacologic product, they have to screen panels. And every so often they overlook one genetic background. Oh, and whoops, it just so happens that that particular ethnicity has a vulnerability in their liver to metabolism of that drug. Wouldn't it be nice if we could predict that before we hurt people? So the possibilities of getting a very genetically representable population of cells of all different differentiation states and organ types, that we could screen drugs across those before we give them to people who could potentially be vulnerable. Wouldn't that be nice to do that with something like vaccines? Identify what kinds of vulnerabilities there might be. Okay? What are the concerns? What are the limitations? Well, it's important to note how they get those transcription factors expressed. They use a genetically modified virus, and they infect those cells with this virus, lentiviruses, retroviruses, and they force those cells to make these transcription factors that they normally wouldn't. So that's not natural, okay? And it's not perfect. So ideally, they would find a way of treating these cells where it's reversible, where it's something like we could turn them on, and then once they're differentiated, we could remove that. That possibility does exist, actually, but it's complicated. We need to make sure it's responsibly used. This is actually a technology that's not really all that hard once you have a basic understanding of this stuff, and that's worrying. Somebody with the means could do all kinds of nefarious and evil things with it. Okay? And again, I brought up Kim Jong-un. He, no doubt, could hire unscrupulous people to do this for him fairly easily. Okay? So cloning humans, that's a bioethical problem. Eugenics is always something that we're concerned about, people selecting certain features and destroying certain others. And if you think that doesn't happen, it does. Okay? There's actually gender selection going on in various countries throughout the world, even though it's illegal. Okay? So it happens. These things happen when there's an unfavorable trait. Okay? Expense and equity. This is not going to be something that's available to Joe Schmo off the street initially. This is going to be available to the most powerful and wealthy people. So there are certainly social implications for that. And this, again, is one reason why it is so important that we have a diverse sort of collection of people who understand this and who are legislating, who actually know it before they legislate. I have to put that out there because it seems like there's a lot of legislation from people who don't know what they're doing. But but there has to be a diverse representation of people who understand this stuff and write equitable law that manages it, and that we're all aware of what's actually going on so that laws are being enforced, because that's a hard thing to do. Okay? Gene editing. Historically, scientists could not control what DNA changes, where insertions happened. So in gene therapy trials, when they try to replace a defective gene, they would basically deliver it with a viral vector, a genetically modified virus, and they deliver it to a human body. Okay? For example, bubble boy. Okay? SCID, severe combined immunodeficiency. People who have this, I say boy, and it's called bubble boy syndrome because they don't live past childhood and they have to live in a bubble. Okay? They have no T cells, no B cells. So they will die, because every sort of microbial insult is very life-threatening to them. So if we could replace the defective gene in that circumstance, wouldn't it be brilliant? And they tried that. And unfortunately, they couldn't control where the DNA inserted. And so when they replaced or tried to provide a healthy version of that gene, it inserted in a bad place, and it caused the development of leukemia, of cancer. And so all gene therapy trials in humans throughout the entire world went on pause for several years after that, with good reason. We couldn't control where it inserted. And if it inserts into the wrong place, it can turn off a gene that puts the brakes on the cell division cycle. Okay? Or perhaps sometimes it can insert into a place and cause the overexpression of something that says, hey, divide, cell divide, and makes the cell divide too quickly. And then you get genetic instability and the development of cancer. Okay? So without controlling DNA insertion, gene therapies are very challenging. CRISPR, Cas9, has revolutionized this. And so basically, CRISPR-Cas9 is part of the bacterial immune system. For a long time, we didn't understand what this stuff was doing in the bacterial genome. You would see these little tiny pieces of viral DNA sequence in a bacteria. Viruses are not bacteria, okay, everybody? So viruses can infect bacteria, right? And they put their DNA in, right? They put their genome in. And these little tiny pieces were being found inside the bacterial genome. And what we found is that those are actually sort of like a matching system, whereby the Cas9 enzyme holds onto an RNA copy of those and goes and looks around. Do I see this? Do I see this? If a virus that has that sequence comes in, it's going to cut that double-stranded DNA, making that virus unable to infect the cell. So it's an immune system of bacteria. It's ancient, okay? So once they discovered how this was working, they figured, hey, we could program this for some human genes maybe. And in fact, that's where we are today. So you can actually select a sequence from anything, any kind of sequence. And you can get it with just the right sort of little loop tail at the end. It will be recognized by the Cas9 enzyme, and it will double-strand break the DNA and cause a sort of repair mechanism that you can then get to put in and control insertion of the correct gene. So you can repair disease state. And that's what I was telling you that Chinese group just did with human embryos. Wow. Okay? Just this week, the news came out. So combined with the DNA homologous recombination, where you put in a correct version, and the repair mechanism naturally selects that piece you put in and fixes it, puts it in the right place, exactly where it should go. Beautiful. So once this came out, holy moly, okay? Wired magazine, no hunger, no pollution, no disease, and the end of life as we know it. We have entered the Genesis engine. Editing DNA is now as easy and cut and paste. Welcome to the post-natural world. So they're expecting that this is completely going to revolutionize everything, and in fact, it's going to be seen as natural or normal to have been genetically modified. Okay? And then this prompted a huge sort of collection of scientists. It was sponsored by the United States, National Science Foundation? I'm not entirely sure, let me see. The Royal Society of London, the Chinese Academy of Science, and I believe it was the National Science Foundation, but I could be mistaken on that. But they all collectively said, we need to meet. We gotta talk, okay? You can change, you can build custom babies. This is dangerous technology. Don't you think so? That's dangerous. I made the joke about like, oh, wouldn't you want bat wings? You tell me somebody in the world wouldn't be like, I want to have a baby that's furry, it'd be cute. There is somebody who would do that. If somebody would do that, somebody will, okay? So this is something that we have to be aware of. It's something that responsible scientists have to have agreements so that crazy, horrible stuff like that doesn't happen, okay? So they got together and they all came to an agreement. We're going to cure human disease. We're going to work on all these interesting projects, but we are not going to affect the human germline. What that means is that we're not going to tinker with reproduction. We will fix existing disease, but we're not going to, those embryos they made, those are not going to grow up, okay? They're not going to make humans designer babies. That was the agreement. Because they don't want to have some massive shift in the entire global human genome. We just don't know enough to predict what we're doing with stuff like that. And you need diversity. It actually is safer for a population of organisms if there's genetic diversity. Too often we see bottlenecking occur where entire populations, the only population of some species that's left, is now extremely vulnerable to something because there's no genetic diversity. So if you start to create designer babies and limit genetic diversity in the human race, that's bad for all of us. So this was a really important meeting. Now I have a little video to show you on CRISPR-Cas9.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise. for example by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
So Feng Zhang is who basically moved this technology over into the mouse. The work that was done first to identify Cas9 and its ability to be reprogrammed, meaning that you could select a DNA sequence, that was actually work done by Emmanuelle Charpentier and this woman down here, Jennifer Doudna. So, naturally, because this is an extraordinary sort of paradigm-shifting technology, there's a patent dispute. Because there are billions and billions of dollars on the line. So Jennifer Doudna at UC Berkeley identified the fact that you could in fact reprogram that little piece of RNA that goes in the Cas9 enzyme, but then Feng Zhang, this brilliant guy over at, I believe it's MIT, the McGovern Institute that just did that video, he is the one who actually translated that into a mammalian application and showed you can do this in mammalian cells. And so there's a big dispute now, and oh my gosh, it's a very big deal. And you will see a Nobel Prize for this process. But the Nobel Committee, it's thought, is waiting because they don't know who to give it to because of this patent dispute, and they don't want to put their foot in it. So they're kind of waiting. I would guess all three of them will probably get it, frankly. But yeah, I guess so. But I'm not involved enough in it to know all of the little political stuff that's going on. But they have to be careful. And this next story I'm going to tell you about will explain why they have to be careful. So let's talk about tissue engineering. With those two things I just shared with you, the ability to take an adult cell and take it backwards to an embryonic state where it's totipotent, okay, can become all different parts of the human body, the fact that we can control the growth into these different tissues, and the fact that we can genetically manipulate it with relative ease. Those are incredibly powerful tools, but now when you add to that the possibility that you can grow organs, so tissue engineering, is the ability to grow whole tissues organized the way they're supposed to be. And that requires a few things. It requires the cells that can actually propagate on the scaffold, the architecture, the structure that they build onto and that they become a part of, with the correct growth factors maintained in the right environment with moisture and with the right percentages of molecules available to them and air. You got to have the right percentage of carbon dioxide and everything. So in the appropriate bioreactor, which of course has to be determined, that can create an engineered tissue. And I'm here to tell you there's two examples that I've come across that I'm like, wow, that's amazing. So when you're determining what to use for a scaffold, there are some considerations you have to make. It has to be biocompatible. It can't be something that the body goes, ugh, get out of here, and rejects it. That's terrible. That's a graft rejection. That's bad. It's for different reasons than standard graft rejection, but that would be bad. It needs to be, and some people will say biodegradable. That doesn't mean you can throw it out and it's going to break down. That's not what they mean in this context. What they mean in this context is that it could be bioreplaceable. So once your cells are established on that scaffold, your cells can go, oh, I'm going to keep regenerating this scaffold to keep it healthy and strong. Okay? Because we always have to sort of recycle the proteins and things that are around our body. The mechanical properties have to be appropriate. Some scaffolds have to be really elastic. Some of them have to be very rigid. Some of them have to be more rigid than others. They have to have certain shapes. Architecture is important. It has to fit in the space that you're replacing. It has to be able to be manufactured. It doesn't help the human race if you can do it once. It has to be something relatively affordable. something repeatable, something that we can acquire. Okay, well, people die all the time. Can we use cadaver tissues? Well, here's a little story about that. Claudia Castillo is a Colombian woman who was living in Spain, and she developed tuberculosis. She's a young mother of 2 when it happened, and she developed a terrible case of tuberculosis. The tuberculosis caused scar tissue to develop and damage to be done to her airway, so you can see it created a blockage. Her choices were you could have a left lung transplant and be on immunosuppressants for the rest of your life, which is dangerous, by the way. It makes you vulnerable to all kinds of things. Or you can have this revolutionary surgery whereby we are going to implant a cadaver sort of trachea, it was kind of, well, bronchus, I suppose, with your cells. And that's what she opted to do. It was a totally revolutionary surgery. Her life was on the line, and she thought, no, I'd like to try that. So what they did is they took a cadaver windpipe, trachea, okay, and they used enzymes to remove all of the cells from the person who donated it, who had passed away. And that left behind a connective tissue scaffold. Those connective tissues were deposited by the cells of the person who died. And that scaffold was perfect. It was just roughly the right size and shape. It was exactly what it needed to allow her cells to implant upon it. So they took some of her cells and they were able to actually implant her cells very quickly, actually, within days, into this replacement windpipe, cut it to exactly the right shape, surgically implanted it in her, and she now is fine. Years out. So 5 years out, I have a paper, and it's on the references if you guys want to see it, but 5 years out, they looked into her prognosis and everything's great. She had a couple of little incidences where it collapsed and they had to put some stents in, but it kept it alive and she's fine. So yes, there was some additional efforts that had to be made clinically, but it worked. Now, here comes the controversy, and I did tell you there was going to be some regarding the Nobel Institute, and this is it. Dr. Paolo Macchiarini is who did this revolutionary surgery, and wow, amazing, but he's disgraced today. He's disgraced because he lied on his CV about where he had worked and what he had done, and he also did some sketchy surgeries on people who weren't sick enough to warrant the risk of what he was doing to them. So he is disgraced. And he's so disgraced also that it affected people at the Nobel Institute who recommended he get hired. I believe it was at the Karolinska Institute in Sweden, which is where the Nobel Prize is given and awarded. And so those 2 people who promoted him or who supported him and encouraged them to hire him, they actually got kicked off of the Nobel committee because of their support of him. So he's disgraced, and he tainted people around him because he lied. And I have to say, stem cell research has also been slowed and tainted by a previous liar. There was somebody in Korea who actually claimed to have cloned different animals, and he was disgraced when it came out that he had lied about some of his data, and furthermore had forced some of his female postdocs to donate their eggs so he could do research on their eggs. Sketchy, crazy crap happens in biomedical research, and that's also why we all need to pay attention to it. Okay? Bad stuff happens. So he's disgraced. Shame. And he wasn't doing any more of these surgeries anyway. So this is the next video I want to show you about tissue engineering, and this is actually the end of the talk, and there's just a little slide at the end I'll show you. But this is actually a skin gun that's developed from the stem cells from healthy skin. Burns are notoriously a difficult thing to treat, okay? And of course, and they leave behind terrible scars. And so if we could come up with some therapies that could repair skin more readily, that would be a brilliant thing. So what they do here is they take some healthy skin from a different part of the body of a burn victim. And this, I have to clarify, is second-degree burns, even severe second-degree burns. Once you start talking about third-degree burns, you're actually getting into the deeper tissues below even the hypodermis, way deep down there, okay? And so you cannot just spray skin and be like, hey, there's skin over your deep tissue, blah, okay? No, they have to actually do a skin graft in third-degree burns still. We don't have that capacity yet. But this skin gun is actually really promising for second-degree burns, even really severe ones. They take the stem cells out of, because you do have a small number of cells that can differentiate into and terminally differentiate into your skin. In your healthy skin, they collect those cells and they create effectively spray paint. And they spray it with a gun called RECELL. That's the one, R-E-C-E-L-L. I only put skin gun at the top there, but it's RECELL. And that's the one that's FDA approved, because this has actually been going on in Europe for quite some time, but our government sometimes holds things up. So RECELL is the one that we have here, and they literally, like spray paint, go all over the burn and the skin grows back. So here's a little video on that.
Skin is the largest organ in the body. And when it's severely burned, it needs to be replaced quickly and massively.
The standard techniques that we have now, that takes weeks, months, sometimes. And the reason why people die is because of infections that develop while we're waiting for the skin to heal.
How are you doing pain-wise?
Pretty good.
Okay.
So if we could find a way to get normal, healthy skin, as much of it as we want, within a week, that's the holy grail in burn surgery.
George Gerlach thinks he's found a way, but he'll need a gun.
We call it a skin cell gun, a device for the deposition of skin cells onto the wound.
It looked like something you'd see in Star Wars.
This is Matt Urim, state police officer from Pennsylvania. He's one of the first people to be treated with the gun.
I attended a July 5th party at a friend's house, and there was a small bonfire. I was standing next to the bonfire, and an individual decided to throw a cup of gasoline on the fire. The worst part was my face. It was the whole right side of my face here, my ear, my neck, and my shoulder, and the entire top part of my arm.
Urim had suffered severe second-degree burns. This photo shows what a second-degree burn looks like.
The arm kind of looked like a piece of charred meat, like a hot dog that was left on a grill too long. The doctor said that I might be a candidate for this new procedure, which is the spray gun, and asked me if I would be interested.
Skin cell spraying is like paint spraying. Just you need a more sophisticated device, like computer-controlled. We isolate cells from the healthy part of the skin, the patient's own cells, which can be taken in a water solution, and that solution is prepared for cell spraying.
Basically, what they were doing was taking my stem cells from my skin and putting them all in the gun and spraying it on my arm.
Scientists have been able to regenerate sheets of skin for decades. The problem is it takes weeks for that skin to grow, and the new skin is extremely fragile. Gerlach's gun solves both of these problems.
It takes like 1.5 hours to take the biopsy, to isolate the cells, and to spray the cells.
This is what Urim's skin probably looked like before treatment. This is what it looked like 4 days after it was sprayed with his own stem cells.
They did it on a Friday, and my follow-up was that Monday, and the burn unit said it was healed, completely healed.
Though the skin gun is still experimental, over a dozen patients have already been successfully treated.
Wow. I saw that, I was like, what? Okay, so I do want to point out a couple things. Number one, they used his own stem cells. Okay, those were some of the few stem cells that we have remaining as adults. But also, there was no scaffold required because it was a second-degree burn. If we were talking about something like a third-degree burn, you'd have to engage 2 different embryonic germ layers of potency, ectodermal and mesodermal. So it's more complicated. But there's promise. There's actually a group that's working on a 3D printer that can actually spray particular different types of cells to try and bring back things like sebaceous glands and hair and sweat glands. Wow. Okay? So the possibilities are there. We're on the cusp. That's why I say we're on the shoulders of giants right now, and we're on a precipice. So these combinations of these therapies are incredible. The requirement for diversity in the scientific community is crucial. Because if you're left behind in what's coming, bad things are going to happen. That's historically we've seen that. Okay? Where certain populations aren't involved, they're more at risk, right? Thank you for joining me today.